Welcome to the German Nagoya Protocol Hub's podcast series, Nagoya Bites. Nagoya Bites is all about helping researchers in the academic sector in Germany with understanding their obligations arising from the implementation of the Nagoya Protocol. My name is Elizabeth Karga, or you can call me Liz. I'm the manager of the German Nagoya Protocol Hub, or the GNP Hub for short, and I'm your host today. I'm delighted that you've tuned in. If you find this podcast useful, or you'd like to suggest a topic for a future podcast, don't forget to let us know via the contact form on the GNP Hub website. You can find the link in the show notes. Today, we're talking about taxonomy, phylogenetic analyses, and sequencing, and how these activities are handled by the new EU guidance document. Understanding what types of research are covered by the regulation is a challenge for a number of researchers and sometimes even for the experts. Here to shed some light on this today is Chris Lyal. Chris worked as a researcher at the Natural History Museum in London for more than 45 years and now is a scientific associate of the museum. So in other words, he's actually retired, but he's still very much active in this space. He's a taxonomist by training and has published extensively on insect taxonomy, the relationship of taxonomy to conservation and sustainable use of biodiversity. Unsurprisingly, he came into contact with access and benefit sharing, or ABS, during his long career and has also published widely on this topic. So, Chris, I'm glad you're still interested in ABS and can be here today. Welcome.
Thanks, Liz. That's great. I'm certainly still working on taxonomy. I've been looking at beetles this morning, which is lovely. But as for ABS, well, this is both interesting and challenging. And it's really critical for our sector as taxonomists. Probably one of the most important things I've been involved in over the last few years has been the CTAF Code of Conduct and Best Practices, since this provides tools that can be used by taxonomic institutes across the EU. That said, there are far too few people working in the science policy interface, and we can't really afford to ignore this. Whether we like it or not, policy does affect science.
So today we're focusing more so on the implementation of the Nagoya Protocol in the EU, and in particular, as I mentioned before, the regulation. So we're focusing specifically on taxonomy, phylogenetic analyses and sequencing, and want to explore whether these activities fall within the scope of the regulation. So in other words, whether these are a form of utilization. Just a reminder to the listeners, utilization means research and development on the genetic and or biochemical composition of the material, including through the application of biotechnology. As research, utilization creates new knowledge about genetic resources. So this concept is a little bit of a grey area. The German Nagoya Protocol Hub has also received a lot of questions about this from researchers who are just not sure if what they're doing is utilization or not. So Chris, in your opinion, why do you think that people struggle with understanding this concept of utilization?
Well, I think the first thing to say is that UN language, and we're dealing with UN language here, is not always clear. And you do very often need a glossary to understand what documents are about. And this term, utilization, okay, the more you look at that definition, the more vague it becomes. So, research and development. Well, we all know what research and development is, don't we? Do we? So, there's a question, does research and development describe only a process where research and development both take place? Or does it apply if there's only research with no development? And that was a question which was addressed quite early on in the development of the EU regulation. And a decision was taken that research, even if there's no development, does fall within the definition of utilization that's going to be used in the EU. And this decision was taken to conform with the definition in something called the Frascati Manual. It's entirely possible that not everybody is familiar with this. And if you look at the Frascati Manual, the term research and development, as defined there, covers three activities: basic research, applied research, and experimental development. So that's the concept we have used for utilization in the EU. It's that research and development, all part of a continuum, and that kind of helps a bit. There are some more elements, and I think we're going to come onto those later, that help refine that down. It is difficult, but it does include just the research.
And taxonomy, for example, is considered a basic and it's usually non-commercial research. There is a bit of a misconception that ABS has nothing to do with this. What can you say about this kind of misconception?
Well, I think first of all, it might be helpful just to tease apart ABS as a whole from the EU ABS regulation, because they're not exactly the same. So any country may choose to exercise that sovereign right and require benefit sharing in return for utilisation or even just for access. Now, benefit sharing is not just about money. Benefits can be non-monetary, for example, taxonomic information about what is collected, specimen data, specimens themselves, capacity building, lots of things. All things that can arise from taxonomic research. Now, this ABS exists outside the Nagoya Protocol. Not all countries are party to the protocol, but non-parties may have domestic ABS legislation, and have had it for many years prior to the protocol. ABS laws in those provider countries often cover basic research as well as non-commercial research. So in other words, researchers may need an ABS permit and a benefit sharing agreement when they collect material from those countries. Now, the EU regulation is there to implement the Nagoya Protocol, which sets in place the way that ABS should work. And part of that is a compliance mechanism in user countries, so that people utilising genetic resources are subject to oversight by their governments. And that's what the EU regulation does. So basic research, it is included in the Nagoya Protocol. So because many countries with ABS legislation include basic research, the EU regulation does as well. So the guidance document is very clear that basic research is included in principle. Doesn't have to produce commercial outcomes to be in scope. And the guidance document does provide some criteria that when applied can exclude some basic research from scope, which we can discuss in a short while. But the bottom line, ABS can apply to basic research. The EU regulation compliance obligations can apply to basic research.
So basic and non-commercial researchers always need to work out whether or not they have ABS obligations. And by this we mean under the ABS laws in the provider countries, but then also whether or not they have additional compliance or so-called due diligence obligations in Germany and the EU.
Absolutely. Now, if a researcher's using material that's already in a collection, then under the EU regulation, they should discover if there are, or indeed should have been, conditions on its use set by the provider country.
Okay, let's look specifically at this example of taxonomy. We referred to it a couple of times already. So taxonomic work is basic research. We were also referred to utilization. Is taxonomy utilization according to the EU guidance document?
No and yes. Taxonomic work is generally not considered to be utilization. So identification, classification of organisms, for example, is not thought to be a form of utilization under the regulation, as long as the research is not focused on the function of genes. So taxonomy is by and large considered to be a step before utilization, that is, as considered by the EU regulation. The guidance document gives something called a litmus test, and it says that users should ask themselves whether what they are doing with a genetic resource creates new insight into characteristics of that genetic resource which are of potential benefit to the further process of product development. If this is the case, the activity goes beyond mere description and should be considered research and development, and therefore falls under the term utilization. So in the case of taxonomy, the document states taxonomic identification of biological or genetic material by morphological or molecular analysis, including through the use of DNA sequencing, is not considered to constitute utilization in the meaning of the EU ABS regulation, as it does not involve the discovery of specific genetic and or biochemical functionality, and it says brackets properties. And that logic is present throughout the guidance document and applied in a number of different sectors. So taxonomy using morphological characters or sequence data would not be in scope of the EU regulation. If, however, the taxonomist starts investigating or using the gene functions, then it's likely that the work would be in scope.
Okay, so one example, I identify a new species. So this would not be considered to be utilization, according to the EU guidance document, correct?
Almost certainly. It makes no difference if the organism identified is a known species or a new species, as long as this does not involve research into the properties of genes. Again, the guidance document is pretty explicit about this. For example, one of the projects that's sitting on my shelf is looking at a couple of weevils that feed on sugarcane. They are pretty well indistinguishable, and at the moment they're sitting under one name. I can distinguish them by looking at a genetic barcode, cytochrome oxidase 1 sequence, and using that to distinguish the two species would not be in scope. There's another way I can distinguish them, and that's because of using the pheromones that they produce, because they produce different pheromones. And that, of course, is genetically controlled. We're then looking at the properties of genes. So using that aspect of the work would likely be in scope. So not a simple answer, but again, there is a way of distinguishing what's in scope and what is not.
So you referred to these DNA barcodes, these sequences. They're short snippets of the genetic information. So if we're using them to identify and or classify the organism, this is not considered utilization.
Yeah, absolutely. The guidance really is clear on this. Just using the sequence without research into functionality is not in scope of the regulation, not utilization according to the regulation. But, of course, it may be covered by the original permit conditions, the pick and the mat. Now, in some cases, these prohibit sequencing or state that sequencing should take place in the provider country. So, merely because it's not in scope of the EU regulation does not mean that there are no requirements on the researcher.
Yeah, so this is an observation that I've also made, that a lot of researchers don't appreciate that there may be ABS obligations in the country where the material comes from, even if the EU regulation doesn't apply to the type of research that they're doing.
Yeah, that's right. So all the time you've got to think about not only the EU regulation, but also, and first, and particularly, about the legislation in the provider country. So always check for the relevant ABS laws and the procedures before you start your research or before you start collecting organisms.
What would you say to people who ask themselves, well, why bother? I'm not in the scope of the EU regulation. Why do I need to go to this extra effort of checking these laws?
Well, and generally, we don't ask ourselves whether we should bother to obey the law or not, I don't think. I hope not anyway. But look, okay, let's be clear. If you breach national laws, there can be results from that. If you're in a country, or indeed out of it, there are sanctions, sometimes criminal sanctions. There are even examples of taxonomists being put in prison for collecting without permits. There are implications about publication. Some journal policies are beginning to ask about ABS compliance before publication. Yesterday I was speaking to somebody who is an ABS consultant to a journal and queries authors about ABS compliance. There have been several cases recently of published papers being retracted after publication because some of the specimens covered were collected illegally. And the government in the country concerned came back to those researchers and said, why is this in print? And quoted a bit of legislation which again had criminal sanctions for doing that. So there's also, of course, potential massive reputational loss. There are examples of researchers being labelled as biopirates in the press. It may be difficult to work again in a country if the country believes you are not adhering to their laws. And of course it damages the reputation of the whole sector. And all the examples I've got are from basic non-commercial researchers. So yeah, there are definitely repercussions let alone the whole ethical bit about complying with laws in the first place, which you really should do.
Okay, let's move on to this question of phylogeny and markers. I'm often asked questions about these as well. They're often used to build phylogenetic trees, and markers are used to delimit populations from one another. Some researchers use non-coding parts of the genome, but some also use markers that do have a function or could potentially have a function. Or a lot of people use SNPs, these single nucleotide polymorphisms, as well. So there are different types of things that can be used for analyses. So could you say something about the use of these markers and whether these are also, or could be, these types of analyses could be a form of utilization?
Sure. So there is actually a section on phylogenetic analysis in the guidance document, simply because this is something that rose as a problem and something that needed exploration and understanding. So the use of markers to create phylogenies and to delimit populations from one another, this is not utilization under the regulation, as long as there is no research into the function of the genes, the function of the sequences. So the same logic as I said earlier. I mean, it doesn't matter if the genes have a function, or even if you know what it is, if that function is not part of the analysis itself. That said, the use and interpretation of the results of the phylogenetic analysis to generate new knowledge could trigger the research being in scope. So the exact transition from phylogeny or taxonomy to utilization really needs to be handled on a case-by-case basis. I mean, it's pretty clear if it's just using the sequences, that's it. But if you're starting to use that to compare something else, then you really may need to look at it more carefully. It's very difficult in the guidance to cover all the possibilities. If you are concerned, then maybe your institution will have an ABS expert, or indeed the competent national authority for your country is very likely to be able to help you on this. They are by and large much more interested in helping people than chasing people. But do take a look at the guidance document. There are some examples given there to indicate possibilities where it is in scope as well as not.
All right, so we've talked a lot about sequences. Sequences are used in these phylogenetic analyses. They're used a lot in taxonomy. So sequencing is ubiquitous in the life sciences these days, and much of modern biology would just not work without it. I know some people still do morphological analyses, but a lot of people are using sequences. So, I mean, there are two aspects to sequencing, and you did mention or touch on this before, these international policy discussions, and there's a lot going on, this discussion around digital sequence information, or DSI. This is a very interesting and also controversial discussion, which we're not going to go into detail about today. As I said at the beginning, we're focusing on the implementation side, we're focusing on the EU guidance document, and now we want to look at basically how the EU guidance document treats these sequences, or this DSI. So what is the status quo, and how's it dealt with?
So the act of sequencing itself is likely to be out of scope. Certainly if you don't do anything else with it, just sequencing, letting it lie, it would be out of scope. But it's sequencing plus the type of analysis that you use is what determines whether it's in scope or not. So, as I said before, just using the sequences, both self-generated and downloaded, for taxonomic purposes is not in scope. But if you use the sequences that you generate for further research, so you start looking at the proteins for which they code, look at the protein structure maybe, look at the function of the genes in a biological context, then you've really moved to be in scope. So again, the sequencing part of it is not, but once you start moving further into functionality, then your research comes into scope.
So it's really the analysis of the resulting sequences which really determine whether or not this falls into the scope of the EU regulation or not. A lot of people these days don't sequence their own material. They send it off to commercial laboratories, for example, to have this done. So let's assume that the resulting analysis does result in utilization. Who's responsible here? Is it the researcher, or is it the Does the sequencing laboratory also have some obligations?
Yeah, this is potentially a complex one, and one you've got to approach with a degree of care. Now, it is discussed in the guidance document. Service providers, people who are just doing sequencing for somebody else, or just doing any sort of analysis for somebody else, because service providers can do quite a lot of things, they typically do not have obligations given a certain set of conditions. And those conditions really should be put in a contract between the researcher and the service provider. So it's very clear who has what obligations. So, for example, the service provider should only perform the activities listed in the contract and isn't granted right to perform any other research and development or exploitation activities. They've got an obligation to return or destroy all the material and the information. And if a copy is kept for archiving, then they've got to really inform the entity commissioning the research. The service provider isn't granted any rights on the genetic resources or rights on the results, doesn't have the right to transfer material information to a third party, and the service requester, that's the person commissioning the work to be done, that requester has the obligation to comply with all obligations under the ABS regulation related to that material. So, given those conditions, and you really need to look at the guidance for the full wording, then the responsibilities stay with the responsible researchers who do the research. Now, we've just been talking as if taxonomists are all service requesters, which by and large they are, but I do know of taxonomic institutions which also act as service providers. So again, the provider has to be as clear in the contract as the requester, so everybody knows who holds the responsibilities here.
So I'm often asked what rules apply if researchers obtain sequences from a publicly accessible database. In research, people might generate some sequences themselves, but then they might use thousands or millions of sequences from something like GenBank. What is it? What are the obligations under the EU regulation if I just get my sequences from the database?
So right now there are no obligations. The whole issue of what's called digital sequence information, which would include sequences you get from a public database, is under intense negotiation. Both CBD and WHO and the Plant Treaty, and for marine areas beyond national jurisdiction. But there's no conclusion yet on that. So right now, you would not have to consider that as utilisation under the regulation. But I'm just going to quote from the guidance document again, because it says the use or publication of such data might be covered by conditions set in the mutually agreed terms, which should be respected. In particular, those who access the genetic resource and obtain sequence data from them should respect the conditions of the agreement entered into and inform subsequent actors about any rights and obligations attached to the data obtained and related to any further uses of it. We're into a really challenging area because those rights and obligations may not be transmitted by the public databases. By and large, at the moment, we don't have anything to worry about as taxonomists, but I think watch this space.
So a little bit of caution is needed because researchers using these sequences from the database don't actually know necessarily if the material that was sequenced in the first place was obtained legally. There may be conditions attached which are not reflected in the database. So there is a little bit of caution needed.
Yeah, and some countries are in their domestic legislation stating their rights over DSI in public databases, and that's not reflected in the EU legislation at the moment. I think just be aware.
It's important to keep in mind as well that some provider countries also scan publications and patents using sequence data to try and detect illegal use of the material or material that may have come from their countries.
Yeah, absolutely. And the more we have open access publications, the more we have open access data, both of which are brilliant ideas and where we should go, the more we can expect people to be looking at them. Which again means we just have to keep our house in order.
Okay, Chris, we're coming towards the end of the podcast today. Thank you for your input. It's been very informative and very helpful. I do have one last question for you. You have been working on this topic quite a long time, and you've been advising academic researchers on ABS and compliance. Do you have any last words of wisdom or advice for our listeners today? I mean, you just said keep the house in order, but do you have any other tips for our listeners?
Okay, yeah, I mean, the first thing to say is that probably most, if not all, of our listeners have been doing this for a long time anyway. It just hasn't been labelled as ABS. So we're not starting from a place where nobody has ever been. We're building on a lot of experience people already have. There are a lot of resources out there, and I know you've given links to a lot of them on the hub, which is brilliant. Thinking of a couple of those resources, well, the one we're talking about today a lot, of course, is the Commission's guidance document. Do read it. It's got a lot of helpful explanations, it's got a lot of examples. It can take you a long way. We also mentioned the CTAF Code of Conduct and Best Practices, which is the only one so far recognised by the European Commission, which is pretty exciting, I think. Now, that was written to enable taxonomists to comply with the regulation and to comply with ABS generally. And it's got some great tools in it, so do get a hold of that, do read it, do look at it, do use it. Think about where to go for help and advice if you're not clear. A help desk, like the hub. Look at your own institution, look at your own networks. A lot of people providing help out there. And of course, your competent national authority can be contacted and asked for help. So, yeah, there's a lot out there. You're building on what you're already doing. Just go find the information, and I think a lot of your questions will be answered.
Yeah, so I think that's a good place to finish. Yeah, there are resources out there, there's support out there, and there are people who can be asked if something's not clear to you. Thank you, Chris, so much for joining me today and providing this insight into ABS, into the regulation, into the research that's covered by the regulation and the insight provided by the guidance document. Also for your useful suggestions for the audience. It's exactly what people need, and it's very much appreciated.
Well, thanks very much, and it's been really great to be able to discuss this. Thanks.
So we've reached the end of today's episode, and it's time to say goodbye. Thank you for tuning in to Nagoya Bites. Don't forget to check out the GMP Hub website for lots of other useful information about Nagoya Protocol compliance. And of course, if you still have questions, you can reach out to us through the contact form. What did you think of our podcast? If you have any feedback for us, you can also let us know through the contact form. And if you have suggestions for future topics, you can leave those there as well. For regular updates on the GMP Hub, you can follow us on Twitter, or you can check out the news section of the website. Keep an eye out for upcoming episodes of Nagoya Bites. Thanks for listening, and see you next time.